Hello, and welcome to our third broadcast of Ferment TV. I'm Kit McDonald, and I lead special projects at Ginkgo Bioworks. I'm the creator of Ginkgo Ferment and the project director of Ferment TV. For those new to Ferment TV, we're an experimental space for virtual gatherings by Ginkgo Bioworks in partnership with London-based agency Faber Futures. We're featuring multimedia storytelling from the world of synthetic biology. We're foregrounding emerging ideas from some of the world's leading scientists, designers, industry leaders, artists, and thinkers at the intersection of biology, technology, and society. You can visit ferment.tv to learn more about what we're up to and see past conversations. Now, before we get going with our conversations for today, I have a few housekeeping notes. This session is being recorded so that we can share it online afterward. We're looking forward to your questions, so please share them in the Q&A box at the bottom of your screen, and we'll explore them together in the last 15 minutes of the session. We'll be sending a quick survey following this session, and we encourage your brutally honest opinions. We're excited by building this unique community, so we'd love your feedback. And finally, stick around until the end, where I'll return to preview next week's broadcast. Now, for today's broadcast, we're focused on the theme of belonging, nurturing diverse ways of knowing and doing. Please welcome me in introducing Dr. Sarah Richardson and Natsai Audrey Chieza for this conversation, from Model Organism to Polycultures. I first heard Sarah speak at the Biodesign Challenge Summit, and I immediately declared her a kindred spirit as she began waxing poetic about how perfect cows are. Of course, she was not just talking about heifers and the like, but also their gut microbial symbionts and the relationships the ruminant system evolved to encompass over the last 10,000 years perfectly. Today, Sarah and Natsai are exploring the contextual advantage of the wild-type organism, pervasive monocultures of land, mind, and body, and visions for equitable biotech in practice and funding. Dr. Sarah Richardson is a computationally inclined microbiologist and science communicator. She speaks charmingly to computers, bacteria, and people. She grew up in West Baltimore, and after training at the Johns Hopkins School of Medicine and the Department of Energy's Lawrence Berkeley National Laboratory, she founded the biotech startup Microbyre. She leads the construction of genomic toolkits for non-model prokaryotes. She uses DNA to train bacteria to do their inborn tricks on command. Trained to straddle disciplines, she has a very unique perspective on the emerging technologies made possible by synthetic biology. Sarah, that is a wonderful, wonderful bio. And today's moderator and production collaborator, Natsai Audrey Chieza. Natsai is a Zimbabwean-born London-based designer, entrepreneur, and storyteller whose practice is deeply committed to nurturing co-designed, equitable, and biodiverse futures. She is founder and CEO of Faber Futures, an award-winning design agency that operates at the intersection of nature, technology, and society. And with that, I will hand it over to Natsai.
Thank you so much, Kit, for that wonderful introduction. And I don't know if you looked at the calendar, but it's actually been almost exactly a year to the date since we all met Sarah last year at the Biodesign Challenge. 17th of June today. I think we met on the 20th of June of last year.
That's a good memory.
Well, I looked it up. I knew it was around about this time. But it's just wild to me that so much time has passed and yet so little time has passed. But Daniel Grushkin from Biodesign Challenge invited us both to give keynotes. And I remember when you sort of opened your mouth to speak, I realized that I'd never felt so validated before in my life, because I think for the first time, I was listening to a scientist who was speaking my language, and a language that I'd been quite coy, I think, to deploy for fear of sort of overcomplicating an already complicated science-design relationship. But I also connected, I think, because we shared something in common. We both work with wild-type organisms that challenge established thinking in our respective fields. And like Kit said, you know, you talked about microbes and how they could talk. that there were ways in which we could become symbionts with them, and that diversity was almost a critical ingredient to expanding the possibility space for biotechnology. I'm really grateful for these kinds of aha moments and what they can catalyze in our thinking, and that they can bring people together in such a way. So thank you so much, Sarah, for being our guest today on Ferment TV. I'm super excited that we can finally have this conversation together. I remember afterwards chasing you and saying, I need to talk to you, but I need to talk to you in a public domain because there's too much overlap. We need to unpack this. So I'm glad that we can finally have this discussion, but also we can share it with everybody who is currently on our live stream today. So the title of today's broadcast is From Model Organisms to Polyculture. And I think that in the short time that we have today, we can try to decode the meaning behind this together. So Sarah, I'd like to start by asking you to share with us the journey that has led you to found Microbyre in 2017, and what your vision is for the company.
You have set a high bar for this conversation, so I hope we can meet it. Thank you very much for the compliments too. I do believe everyone can understand science. It is our job, if we're scientists, to get the idea across. And it's supposed to be multidisciplinary. It's an effort. It's an ecosystem. But I started wanting to work with weird organisms when I was in the 7th grade because I really watched too much Star Trek and I wanted to meet aliens. I really believed that first contact was the coolest puzzle anybody could solve, just figuring out how to talk to another organism. And the tragedy of not being sent into space to do it was allayed by the discovery of bacteria. My discovery of viruses and bacteria. Microbes are the weirdest form of life around. They're just really, really cool. And then I discovered that most of them were considered unreachable. They're just out there doing their thing, and they don't really care or know about us, and we don't really reach out to them. We study them a bit, we're aware they're there. So it wasn't something that was easy to do in grad school, but I worked a lot on, I kept focused. One of the reasons I went to medical school was I thought I would be really interested in the whole microbiome component of human bodies, and how if we could be friendlier with it than just it's there and sometimes we eat yogurt, that we would be able to address a lot of the human ailments that cannot really be tied to our own genetics. But I really fell in with the industrial biotech side instead, where the larger questions were those of climate and agriculture, biomanufacturing from clean sources. And that's why I went to the Lawrence Berkeley National Laboratory with the Department of Energy. And I told them I have to work on non-model microorganisms. But that's still really hard. We're missing a lot of data. So Microbyre was started as an automated attempt to gather a lot of the data you need to be able to confidently approach independent microbes and convince them to join us in biomanufacturing. Yeah.
So maybe it might be really useful for you to explain to our audience the difference between, what we mean when we say model organism and,
Sure.
and what we mean when we say wild type.
Sure. We usually use the term wild type to refer to an organism that has been recently isolated or has been, say, unaltered by selection by human hands. And a model organism is one that we use as a stand-in to represent a clade, a group of organisms. So there's models at every stage for biologists to study. I think high school students might be familiar with the frog or the fetal pig as a model organism for physiology. In microbiology, we tend to use a lot of lambda phage as a virus, E. coli, Escherichia coli, as a bacteria. We use Saccharomyces cerevisiae, which is the same yeast everyone is doing for their baker's craze during the shelter in place. Moving up, we use Arabidopsis thaliana for plants, and humans use mice as a model organism for pharmaceuticals or monkeys for certain things. And these are just meant to be stand-ins for other organisms that are more difficult to attract, more difficult to keep, or unethical to test on directly, like humans. So the act, though, of picking a stand-in sort of biases you or limits some of the understanding or testing you can do on the wider range of organisms they're meant to represent. It's a trade-off.
That's really helpful. Thank you, Sarah. I guess I started working with a wild type organism because it was sort of the low barrier of entry, I think, for a non-scientist to be working with laboratory protocols. So I remember when I first started collaborating with Professor John Ward at University College London, he gave me an organism, Streptomyces silicolor, found in the soil. And he said, I asked him, oh, where did you get it from? Where does it come from? Because obviously he got it from the freezer. But I think I was asking about provenance, right? Where do these microbes live? And then I've been obsessed with that question ever since.
It's an excellent question.
But I remember just being like completely blown away when he said to me, oh, I got it from my cactus plant. And I was like, wow, okay, teach me how to do that so that I can understand and begin to contextualize this organism.
I think that's a thing. It might have come easier to you from a different background coming into science, especially for model organisms. Their context is we use them. They're what we're trained on. But if you really want to branch out beyond them, or even to deeply understand a model organism, you need its context. You need to know where you got it. And if there's a function it's fulfilling, if there's something it's making or doing, and I believe for silicolor you were concerned with the dyes, the pigments that it can produce. In my view, it is motivated to produce a pigment for a reason. And that reason can sometimes be deduced by its context. Who is it living with? Where is it living? What temperature ranges are happening there? What challenges is it facing? What benefits does it have? All of these help you understand the organism in context. And then if you are going to model it or approach one wild type and get it to come into the lab and become a model organism, that context is so important to caring about it. Mm-hmm. You have to care about these things. They're motivated.
Oh gosh, care. I guess that leads me to sort of my next train of thought that I have. And it's really based on the fact that design as a discipline is shifting. I think for me personally, I've watched over a couple of decades biodesign, the design of, with, and from biology become a multidisciplinary field. I think that's in ascendance, and it's really starting to push the boundaries of what constitutes renewable materiality. And I think that much of this shift in culture has been driven by a curiosity for experimental practices. Certainly for me, that's how it began. But also this recognition that petroleum-derived materials are an invasive species. Materials like plastics, right? They're in our land, they're in our air, they're in our water. So I guess here I'm starting to situate oil as a monoculture material system that governs, or that is governed, that governs basically what is made, by whom that is made, and the sort of life cycle it inherits because of the logic of the system it belongs to. And I have this idea that, right, and I think we all do, that divesting from fossil fuels to reach net zero emissions puts even greater pressure on our need to find alternative material systems. But it also opens up the sort of opportunity to start thinking about what divestment from the monoculture that the oil age reinforces looks like, right, should we choose to. So there's a possibility, which I think that you represent, not only to decouple materials from petroleum origins, but to start to think, to speak and do design and science with a sort of polyculture mindset. So with that in mind, I'd like to ask you how you've seen monocultures present and manifest themselves in science. What are the sorts of implications of that, and how we might start to consider or reconsider what an alternative space looks like?
It's always been striking to me how we got to, in biology, say model organism. It's similar to how petroleum sort of took over. I think it became, you described it as a model or as a monoculture. My understanding is petroleum took over because we found it was a great source for energy, particularly for military applications. And once you've built the infrastructure for working in petroleum, all of the other benefits like plastic just come easily off. They're subsidized by the massive benefit you get from the primary use. And similar to model organisms, once you have established a system that everyone intends to use, all of the tools work in that model organism. And so those monocultures sort of lock us into a certain way of thinking. It's very convenient. It's the convenience of it, the immediate convenience. But they're also limiting. There's things we cannot do with petroleum, like save the planet. And it's also interesting to me, you can see both of these things arise. You can see the monoculture arise in the history of microbiology. It was sort of an accident. It was not guaranteed. It was not for E. coli as our model organism in microbiology was not guaranteed to have been the one. If you go back and look, there were a bunch contending for that position. And before petroleum hit, we used biomass exclusively. Every organism on the planet channels through carbon, and most of the ones we care about channel through biomass, that agricultural residue composts. And we used to do that too. We took agricultural residue and burned that for fuel. We made all of our chemicals from it. Our plastics we made directly from wood. And we shifted to a monoculture, and it sort of overwrote what we remember being possible. So now again, biomanufacturing or coming from biomass directly seems novel or exotic or like the next stretch to get to. So I think that when you see something amazing pop up, and then there is a righteous and understandable doubling down and infrastructure build around something that is a leap forward, it's valuable to keep the skeptics or the alternatives warm. Mm-hmm. Because the limitations haven't been found yet. Where you're making massive strides in microbiology or in manufacturing, and do we still remember how to do it the other way, or do we remember how to repeat it? In the case of model organisms, for me, it's more of a question of how do we take all the lessons of how we made E. coli so useful and powerful, how we brought it into our fold and domesticated it, and now take that to organisms that can do things E. coli cannot do. And for petroleum, it should be a similar thing. How do we go take the lessons of harnessing that and building that infrastructure and go build one for biomass now? How do we make biomass as easy to move around as commoditized? Even your compost and your lawn clippings should have a value. How do we do that after we've done it for petroleum? And if we adhere too much to that monoculture thinking, that we have these experts, we have these opinions, and they're very loud and they've succeeded a lot, not hearing the minority opinion slows down the process of building parallel or even superseding infrastructures. Does that answer your question?
Yes, it does. But I'm wondering if maybe I know too much, and so if we maybe back up a little bit and refocus. I think a lot of our conversations we've had about the problem with the monoculture is also tied to scale, right?
Yes. Yeah. In fermentation, scientists and industrialists have tended to like monoculture, and I think that's tied to a notion of control. And even in design, we like control. We tend to think of the things we are using to design our processes, our homes, as controllable. Steel does not have an opinion about how you cast it. It has properties that we respect as we do design, as we build things, but it doesn't change. Mm-hmm. And it scales well. We know the limits of its scaling. But monocultures, I mean, we don't build our houses entirely of steel either. Even construction uses multiple materials. So when you're looking at massive fermentations from a single organism that has its own motivation for doing anything, the scale has been hard to come by. And I think it's also an important lesson. At least I'm a biologist and I think of everything in terms of biology, but there are no monocultures in nature, not a single one. There are none. When you think you found one and you scratch a little deeper, it's not a monoculture. And it seems that when you have something that's alone and trying to do everything for itself in its niche, you're going to need to split up and take on different jobs just for your own survival. And in a massive fermenter, it's no different. The biggest fermentation people tend to do is treat our own wastewater. And you cannot do that with a single organism. It is a complex mix of organisms that interact and take on different duties as time goes by to get all of that done. And that is so scalable. You can put as much waste as you want in a pile, and in time, it will come down. But we have no control over that. We sort of cede our control to the outside bits, how big it is, how fast it stirs maybe, but what they're doing, we let them handle that. Mm-hmm. And I think that moving from monoculture to polyculture is going to take some practice in accepting that we don't control, and you cannot control biology or nature. You have to work with it. You have to design with it, not at it or on it.
And you've talked about that. I mean, you use some incredible metaphors to describe this. The one that comes to mind is the recipe book, that nature is full of all of these recipes.
It's hard for us to even comprehend how many different recipes there are. Some of them you don't get to look at them until a certain conditions are met, but they're really useful. Some of them you don't know that they'll ever be used because there's no way to read a DNA sequence and know exactly what the recipe is, what the ingredients are, what the conditions are. But our planet is a massive library of cookbooks where every organism is a collection of recipes that are a response or were crafted as a response to challenges. And so the organism has this whole repertoire of tricks to pull out that it might only need once in a lifetime, that it might need every day. And they are so much more complex and effective and efficient than things we have been directly designing in response to what we've observed from our model organisms. And polycultures of these recipes, they work so beautifully together. They enable all life on Earth. That bacteria specialize in pulling nitrogen straight from the air and feeding it to our plants so we can feed. That they cycle phosphorus, they cycle sulfur. Our life is a symbiosis with their life. And we can't do what they do. We don't have the recipes. So we can't, it's really hard for us to invent recipes too. We've been trying, but again, that goes into a sense of control as opposed to context and motivation. So I think we're not done harvesting from the library, from all the cookbooks, but it is so much easier to assess a recipe or to follow it where you found it. Mm-hmm. That's why I think it's really important to get into or out of the monoculture where the tools are there to say, let's test this recipe in this context. When any chef will tell you the kitchen matters.
It's so interesting that you used the kitchen, the recipe, that way of thinking about it. I remember when I first started working independently in the lab, I had no frame of reference for what I was doing that lives in the scientific body of knowledge and thought. I had to work instinctively to figure out what happens if I do it like this, and listen to and observe the process. And my process of documentation is probably not very scientific.
You were writing it down. That's fine. That's
I was taking pictures and I was exhibiting. And my collaborator John still keeps saying to me, "When are we going to write that paper?"
Yes. Yes. And you must publish or perish.
I must publish or perish. Whereas we construct and then we disseminate in lots of different ways, not least in exhibition. But I was just really struck at how I started to describe what I was doing as, yes, here's the recipe, and yes, here's the protocol or the method, and you start to cook. You know, I use all sorts of metaphors around cooking in the lab, and when you see labs now entering creative education spaces at the Bartlett, I think Central St. Martin's has what one would call a proper lab, but it feels like, right, this is the kitchen. Where's your incubator, the warm room? Where's the autoclave? And for many students, that has been the pressure cooker, and that is that immediate way of accessing what's going on. I guess the challenge ahead is how you take that experimental practice and start to do what you're doing, which is how do you scale this for industry? How do you take that intention? I think we talked about this yesterday, and start measuring some kind of impact with that.
Yeah, one of the hard parts, or at least for me, was realizing that, you know, ideas are great, execution is everything. And there's two fronts that I have to work on to change things that I want changed. And one is the communication, the hearts and minds. And analogies are so important. So, for example, getting those students to be more relaxed, grasp what was happening, and be able to interact with it. And so jump from the kitchen to the laboratory. It's an extremely effective analogy. You follow steps, you pay attention to amounts and timing and order. We also have to be aware of when those analogies start to fail. None of the students should by any means put anything in the laboratory into their mouths because it is not actually a kitchen. So some of the analogies in synthetic biology are extremely effective for education, but start to fall apart when you try to scale or take it to industry. And that's one of the communications I'd like to sort of change, or make everyone aware of the limits of that analogy. And again, it comes back to control. When you analogize bioengineering to electrical engineering or computer science, you are bringing in, you're importing a headspace or a pragma of control that is not feasible in bioengineering. Bioengineering should be husbandry, not control. But for the other battle to be fought is to prove its effectiveness, to prove it by being able to generate value measured in capital. And there is so much value being generated already there, but again, there is a narrative disconnect, a vocabulary disconnect. There are many, many companies that are thought of as chemical companies that are doing what they might say biocatalysis, because that's how chemists speak, but they are using microbes in fermentation. It is, the value of biotechnology should not be in question. I mean, when I say biotech, many people think medical biotech. Of course, no one questions that. Industrial biotech and the deployment of synthetic biology, extremely valuable, but often under different names. So that reminds me of a monoculture, which you alluded to, as publishing. Publishing is meant to be in this format: abstract, introduction, method, results, conclusion. And you were showing your work by going with exhibitions and using a different way to do it. The monoculture of science communication works this way. That locks some people out of knowing what's going on. It makes some of the very awesome discoveries very difficult for other people to grasp and build on, particularly from outside of the field, where you're a very smart person, you can read literature, but this literature is completely impenetrable to anybody who doesn't similarly write papers in that field. We really need to break up, have multiple methods that are respected, and scientists are rewarded and motivated to write in more narrative structures. I was wondering about this bacteria, and so it's so much easier to understand for many people, including me. I would love to write all my papers as narratives.
Actually, I once wrote a paper with John, and it kind of has that la 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 la. And it's amazing because it's like, that's John speaking, that's me speaking, but we found a way.
There's no reason not to be able to do both.
Yeah, exactly.
It's a diversity. It's the ecosystem. It covers the spread.
So I wanted to go a little bit deeper in this metaphor space, and especially since you bring it up, Tom Knight, who is the founder of Ginkgo Bioworks, and you have this amazing conversation that's published in Ginkgo's inaugural issue of Grow magazine, and you sort of disagree quite beautifully, quite openly in your conversation on how to describe synthetic biology, and therefore the framing, the terms of reference for how we go about designing with living systems is sort of implicated in that. How have your ideas and indeed your vision for Microbyre been met with resistance, right? Either in the academy or in startup land? Will we ever be able to tell who was right?
Yeah, I think right. Well, I'm a biologist. I don't think there's a right in biology. There's sort of you live or you die. That's evolution. It's a very, the planet is a harsh mistress. So you try a lot of things, and something that works, it might not be right, but it's effective here and now. And that's how I try to measure things is, are we moving the goalpost to more difficult things? Not are we right, but did we get past that hurdle and stay alive? So the main resistance to what we're proposing to do in this format is just that it's too hard. Mm-hmm. People see value in it. It's clear that nature sees value in it, that has been a way to move the goalposts and stay alive, is cooperation, symbiosis. None of us would be here without the bacteria in our guts. It just works. But it is widely regarded as too hard, too difficult to do, maybe on these timescales, and in some cases impossible. That just some things are too weird, too difficult to bring into the lab. If you think about them from, again, that monoculture lens, we grow E. coli this way, we have all the tools to do it this way. Anything that's outside of that and we'd have to build new tools for, that's an investment in infrastructure or time that we have deadlines. It's too researchy. Those are the main forms of resistance we meet. It's the time scale and the capital required to build an infrastructure that doesn't center around an existing model organism. But I have to say, nobody that I've met thinks it would not be useful. They mainly think it's not proximal or practical. Does that make sense?
And practical based on which value drivers? I know we've talked about investment before, and the culture around investments.
Yeah, it's some of the venture capital models that take flyers on small companies like mine. It's difficult to point to other companies, and it's difficult to fund something that appears to be an R&D company. I don't see ourselves as R&D. You can't make money on R&D. That's too risky even for VC. I see ourselves as engineers. We've used the term scientist to describe me, but I have never really been a scientist, not honestly. I really struggled in grad school because it turns out I'm not a scientist. I am an engineer. I don't want to discover the basic rules of things. I want to apply the ones other people found. Cool. I totally support basic scientists because I need them, but I am not one. So I view this company as an engineering effort. We are automating collection of data to reduce the risk and the time frame involved in approaching of outside organisms and bringing them into the fold. It's a progress we're saying can be made more rapid. However, if you're on a model where it's extremely cheap and effective to hire a bunch of software engineers to sleep on couches and their overhead is basically laptops, a company like mine looks like a much longer investment to return with much more capital cost. So we need centrifuges, we need mass specs, we need all this equipment that a startup company does not need. So there's VCs out there that get this, have longer time frames, have the capital requirements, have the patience and the experience. But then a lot of them are for clinical biotech. They say biotech, but they're mainly focused on clinical biotech, which we're not. Where I'm an industrial engineer, I try to be. So it's an interesting needle to thread to say we can create value. It's going to be value in this space, and it's going to take a little while. But there are VCs out there, but still, to validate their chance on us, we do have to produce a capital value. In addition to all the goals we have for climate remediation, for bioremediation, we have to make money to survive long enough to do that, to not be right, but to be effective.
Mm-hmm.
Did that answer your question?
That. No, absolutely. And it makes me think about this, something that I've been thinking about a lot. How do our sort of interventions become interventions that are also equitable? And I don't know really, I mean, it's a very wide question. Are we talking about how this is funded in the first place? There's a lot of resistance around an economic model that sort of the oil industry is benchmarked on, that creates the world that we live in. Is it possible to use that same, pardon me, model to challenge and to build better?
Oil sets a bar. In order for biomass to take over, compete, or even run side by side, we can't just match the price of oil. That's not enough of a motivation to the human organism to switch over. We have to beat it. We have to beat the price of oil. We have to play it by its own game. The benefits of oil outweighed the benefits of biomass with our technology 50, 60, 70 years ago. And now we have to make the benefits of biotechnology outweigh the benefits of petroleum. And we all know that for clean energy, net carbon, and emissions, it has the capability. But human organisms are motivated by capital gains. So the same way I try to figure out what motivates a bacteria so that it does what we need it to do together, we definitely have to cast engineering and biotechnology in a way that motivates people. You cannot control an organism. I cannot control people, and I cannot control bacteria. I figure out their motivations and work with them. And if those motivations don't line up, you have to find a different organism. So, you know, Earth will survive even if we don't. And there will be another organism.
That's the moral of the story.
But I think it's stronger we have a better chance if we all know this, we work together. That designers, engineers, architects, construction, all of these different disciplines work together, and then we all get to stay warm and dry when it rains. And we're not going to be able to approach bioremediation, conversion of biomass efficiently to beat petroleum if we're stuck in the monocultures of thought, of labeling, if we call it synthetic biology and not also molecular biology or bioengineering, if we don't understand that all of the successes of one are actually also the successes of the other. And if we don't bring in more organisms to work with us the same way we did for agriculture. I mean, can you imagine a diet of only broccoli and kale? We don't even have monoculture in our crops. So it's really, really important that we are motivated properly, but also that we tool ourselves up properly with diversity. And I really believe that goes all the way down from bacteria all the way up to boardrooms. It just gives you power. It gives you flexibility, and it gives you strength.
Wow. Yes, more of that. I had another question, but I forgot, that relates to exactly what you were talking about. So I might come back to it. You have recently been awarded the 2020 Association for Women in Science Next Generation Award. So congratulations for that.
Oh, thank you.
And we also discovered that in 2015. Oh my God, another time frame. The SynBio Leap Fellowship. You also were awarded that, and the SynBio Leap Fellowship is really for leadership potential and the vision that you represent, I think, for shaping a future in biotechnology. I've been thinking about leadership a lot. The fact that we have people in power, but who demonstrate no kind of leadership to bring any kind of unity. And I'm thinking in a sort of post-COVID-19 world, how do we need to start to really be intentional about leadership? What do you think the biotechnology needs to throw its energy behind in terms of leadership, and how do we foster that? And I say that because I see you as a role model, not just for budding scientists, but for
or engineers.
or engineers. But literally, like, my jaw dropped when you spoke last year, and I thought, I have, in all my years studying architecture and material futures and doing the work, that I've never had a role model, and like, you were it in that moment.
I am really flattered. I think a lot of people don't get training in leadership. We might get training in management if we're lucky. Otherwise, we're sort of thrust into these roles. The only thing I try to remember about leadership is the same thing I try to remember about motivating bacteria, which is to understand their context and respect what they do. That's it. I think that's what biotechnology needs to do in the very technical sense, is respect the abilities of all the organisms from which we're deriving inspiration and work with them where they are. And then you step back and let them really tell you what needs to be done. That's been agriculture, is you say, you're great at this. How far can you take it if I take care of you? If I provide you the resources and the protection you need to really be a star, how big can you make that ear of corn? And then I look brilliant. It's really all the people that I have a respect for, and that I can say, you are going to make this happen. I can't do it. But if I empower you, you can go make this happen. You have all the skills and the understanding and the context to do your passion here and make a difference. So biotech needs to also respect the other silos. The biotech itself can be described as an amalgamation of, say, computational, computer science, informatics, chemistry, biochemistry. I mean, we can split all the subdivisions up, but there's a bunch of other silos that it either hasn't penetrated, maybe doesn't respect, or fears. Things like regulation can be really fraught for a lot of fields, and that communication to that other silo can be scary, difficult. And I think biotechnology can take leadership there, because every person on Earth is using biotechnology and really respects it, really loves it. They love medicines, they love food, they love their pet dogs, which I think is the best form of biotech, is getting a wolf to be your best friend, and even maybe a cat around, maybe. But that we really need to model that respect for other fields and how they enable us to go do our bit. And that's leadership, is to say everyone has a role to play, and those roles are not low or insignificant or not worthy of respect or pay. That the whole symbiosis keeps the energy going. It keeps the people going. And that's leadership, no matter where it is just saying, you can do this and we're all going to benefit. And I'm going to give you all the credit. That's a good one. Make sure people get the credit. Yeah, it's an ecosystem, right? That's what I've been saying. We all everybody's got a part, and respect all the parts.
That's really important, and I think that's something important for certainly designers who are really digging into this field, or fields, I should say, to understand where design is an incredibly helpful toolkit.
Keep plugging. Keep making the scientists and engineers tell you both why they're not getting it, like, really, and to try and redescribe what you're doing, in each other's languages. One of the reasons I think I've had a bit of success was because when when I was in graduate school, I was offered a fellowship from the Department of Energy, and the goal of that fellowship was to make me learn the languages of disciplines that I otherwise would never have learned their languages. I might have used their tools, I might have worked side by side with them, but I would not understand that when they said word X, it meant Y in my field. And so sometimes it's really difficult when we don't have that mutual respect or empathy, but you are putting a lot out there by going to scientists, by saying, let me join your lab, let me see where I can bring my skill sets in. And it's on us to come to you right back there and meet at that intersection and see how we can hook up this symbiosis. And unfortunately for you, this is true for many fields. I'm not trying to call out scientists, but you might have to knock a little harder and ask slightly different questions to get us to come to that table and see your value. That is the Earth's problem right now, is leaders don't necessarily see the value in everything or everyone. And so sometimes we have to knock some heads, knock on doors, be really persistent and annoying, and keep trying new people until we find someone who goes, oh, wait, that's actually really useful. So keep trying. I really respect the effort. It's so easy to get discouraged when you keep getting that signal that your skill set and your passion have no place here. And I mean, scientists are getting discouraged by that too. So it should be a feeling we can all rally around.
No, absolutely. And I think from my own experience, I was incredibly lucky when I first reached out to Professor John Ward, that he was just a really curious sort of old school scientist who to this day always says to me, I keep my bench. He's head of department. I keep my bench because it's the only way I can think. He's still able to do whimsical things at his bench, and I guess in some way I was part of that whimsy because I shared his bench for a long time. But that openness and that willingness to allow something else to happen, and to let it sit with you and sort of marinate.
And sometimes to be disabused of our notions, and sometimes to do the disabusing.
Yeah. It's really important. Okay, so I really want to turn to the audience for some questions in our final few minutes. Oh wow, this is a good one for you, Sarah. This is from Jose Cabezas. The question is, the unexplored genetic landscape on Earth is enormous. What is the most exciting non-conventional microorganism you have worked with? And what are your hopes for the future in terms of domesticating these microorganisms for unique functions?
That's a hard one to answer because I love them all. Yeah, they are all amazing when you get them in their element. But I think one of the coolest ones, it's easy for people to go look up, and there's videos of it, because let me be really clear, I am not the discoverer of these things, not yet. So many microbiologists before me and currently go specialize in studying and observing these things. And so it's really important to know that I am not the sole source of information or even the deep source of information on these. My main concern is breaking them. So, taking that stallion and turning it into a horse we can saddle up. But one of the coolest organisms, I love to tell their story, is Myxococcus xanthus, Xanthus. This bacteria lives in the soil crust. It goes and eats E. coli. So kind of, if you're into killing pathogenic E. coli, that's a plus. But it does it by bumping up against E. coli. It has these different kinds of motion that help break open the E. coli. When it was first discovered in the 1920s, it was thought to be a fungus because it has the largest genome of any known bacteria, and it grows in complex structures. It makes differentiated structures. Half of them die to protect the other half that they can go sporulate. It's just unbelievable. And industrially and in biomedical wise, it makes all these compounds to communicate with itself. You think they're just individual organisms that seem they should be just selfish, but they've managed to cooperate and communicate with each other with these signals. And those signals, because they're telling each other when to stop and start growing, they can be anti-cancer drugs. They make antibiotics. They make all kinds of things that help them survive in a complex, competitive environment. And they're just so cool. They're pretty different to culture in the lab from E. coli. So it's a really good example of assumptions not carrying through. Even though you find E. coli in them in the same environment, they require very different conditions in the laboratory. So that one is absolutely my favorite one to communicate to people. Go online and find a video of it eating E. coli. And then any bacteria that makes magnets is pretty cool, because why are you doing that? What is your motivation? People have theories about their motivation, but man, these make magnets. They're perfect magnets. I could go on, but I think there's probably more questions.
It sort of reminds me of plant tropisms and how different plants sort of respond and grow in different ways, including magnetism. That's really, really cool.
Wild recipes.
On recipes, what sorts of tools do we need to find these recipes? Maybe we can go into that a little bit.
So we're using a lot of automation where we can try making different environments very quickly. So that helps us see the responses and suss out, get a guess at which recipes a bacteria has. Can you survive in this condition? What are you making in that condition? If you cannot observe their function, their life, their responses in many, many, many environments, it's really difficult to know how to motivate them or what they do under pressure or under joy. So that's a tool we have. I don't have to build it. We just have to organize it, which helps when you speak multiple disciplinary languages. But we have all the tools to do this. We need to arrange our motivations. Does that answer your question?
Yes, it does. And then maybe one question from Theo, who asks, what approaches are being taken at the moment to scale up polycultures for industry?
I know, as I mentioned, the biggest polyculture, can you call it industry, is wastewater processing. That is a massive polyculture. I think the next biggest one is probably cheese production. It's not just one bacteria. You put cultures in, you put mixed cultures in to make your cheese and yogurt. So I think there's a lot of models for that, but I'm not sure in all of our chemical fermentation or beer or bread making that we're really interested in too much polyculture. Not yet. It needs to be demonstrated to be as valuable, more than as valuable for the current processes. So it's a risk. It's a risk to take on to say, let me try this. That's why I think the efforts right now are much more preliminary. We need to demonstrate not exactly controllability, but reproducibility and reliability, which means figuring out who could work together productively. And again, that feeds back into the response curves of organisms under different conditions. It's difficult to bring a functioning ecosystem into even a laboratory and have that whole thing keep going if you don't understand atomically how each thing is going to respond and what they need from each other. Same as leading a team as a manager. How are they going to work together and how do you scale that up?
Yes. One more question I think we have time for. This is from Joel Clote. He asks, could the process of domestication suppress the interesting characteristics of one of these wild bacteria?
Oh, absolutely. I mean, one of the interesting characteristics of wolves that we suppressed was their ability to work without wolves. Dog packs are hard to come by in domesticated dogs. They tend to look to people. It depends on what you think of as interesting. I try to think of it less as keeping the interesting as keeping the functional. So we're going to lose a lot of stuff with domestication, if anything. And the main thing we're looking to lose is independence. That we're going to trade them shelter and food and safety in whatever group of them we have for their abilities to live independently. They're going to we're going to want them to lose the ability to feed themselves entirely from whatever carbon sources we give them. We're going to want them to really like living with us and be rewarded by it. So in that sense, yes, there's some interesting things they're going to lose in the process of becoming a domesticate. But the nice thing about biology is we're not going to lose the wild type. We can keep the wild type and just help them focus on this or that. But yes, domestication and bioengineering has to be a subtractive process. You have to take something away in order to have it specialize even deeper in your industrial context. But I try to think of it as, you know, we're focusing on something interesting and it's a trade-off.
And I suppose the benefit of molecular engineering is the time scale that is at play. I think I've just lost my sound, but I'll just say this and I'll figure it out. But the time scale that is at play when we engineer as opposed to domesticate, at least the traditional ways in which it's taken thousands of years to domesticate anything, right? Hi, Kit.
Hello, I'm back. It appears that we lost Sarah. I was really hoping to get some final thoughts from her. But quickly before we wrap up, maybe I'll buy some time to see if she can log in by telling you all about next week's broadcast. Next Wednesday, we're going to be featuring Dr. Alondra Nelson, the president of the Social Science Research Council, and Harold F. Linder Chair in the School of Social Science at the Institute for Advanced Study, and Dr. Joan Donovan, the research director of the Shorenstein Center on Media, Politics, and Public Policy. They will speak to Christina about racism as a public health issue and the social dimensions of pandemics. Natsai, what you and Sarah are both saying about the language and metaphors and different narratives that go into this cross-disciplinary work is so poignant, and I think it reiterates the importance of having these types of conversations across the disciplinary divide, whether it's between designers and engineers or social scientists and different researchers. So I think it appears that we've lost Sarah. So
I think maybe we can wrap up and we will reach out to Sarah. You guys had such amazing questions that I'm sort of looking at now that sort of came in right at the last minute. And Kit and I have talked about how we'd really like to hold on to these questions because I think that they are still relevant relative to the rest of our programming. And we want to find ways to pull them in.
With that, I want to thank you, Natsai, as well as Sarah for this marvelous conversation today. From the Ginkgo Bioworks team, I'd also like to extend my thanks to Christina Agapakis, and on the Fit for Futures team, Laura Vent, Joanna Mann, Camille Thierry, and Magda Ovmalko, and of course our graphic designer, Marta Ubers. So thank you so much, everyone, for joining us today. Visit ferment.tv to subscribe to our newsletter and find out about upcoming broadcasts. Take care.